This song's called Liars, Cheats, and Crooks. You want me scared, you want me weak, you want me brain dead and asleep. You want us trapped while you all laugh behind the scenes. You want us sick, you think we're dumb. You want us blind and you want us drugged. You want us poor while you get more of everything. But you don't get to tell me what to think and what to do. No, you don't get to tell me what is true. 'Cause you're just liars, cheats, and crooks. Change the rules and you burn the books. And so I don't believe a single word you say. You're all liars, fakes, and cons. Want you out and we want you gone. So don't believe this time you'll get away. You want us tricked, you want us numb. You want us scared and you want us stung. You want us shot and you want us bought in every way. You want our minds, you want our time. You want us framed up in your crimes. I hope you know that it's time to go and we're taking names. 'Cause you don't get to tell us what to think and what to do. You don't get to tell us what is true. 'Cause you're just liars, cheats, and crooks. You change the rules and you burn the books. And so I don't believe a single word you say. You're all liars, fakes, and cons. Want you out and we want you gone. So don't believe this time you'll get away. 'Cause we see la 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 all your lies. La 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 all your lies. You got it. You don't get to tell us what to think and what to do. You don't get to tell us what to think and what to do. You don't get to tell us what to think and what to do. 'Cause you're just liars, cheats, and crooks. You change the rules and you burn the books. And so I don't believe a single word you say. You're all liars, fakes, and cons. Want you out or we want you gone. So don't believe this time you'll get away. 'Cause we see la 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 all your lies. La 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 all your lies. Yeah. We see la 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 all your lies. La 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 all your lies. You're just liars, cheats, and crooks. You change the rules and you burn the books. And so I don't believe a single word you say. You're all liars, fakes, and cons. Want you out and we want you gone. So don't believe this time you'll get away. 'Cause we see la 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 all your lies. La 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 all your lies. Louder. La 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 all your lies. La 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 all your lies. We see la 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 all your lies. La 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 all your lies. We see la 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 all your lies. La 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 all your lies. One more time. La 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 all your lies. La 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 all your lies. Perfect. Thank you so much. I'll see you a little later. Thank you all. You're beautiful people. Thank you so much.
Tonight on VSRF Live, Steve Kirsch welcomes psychiatrist and molecular biologist Dr. Andrew Kaufman, who has risen to fame during the pandemic for his unorthodox views on the existence of viruses.
There was just the appearance of devastation, like changing the numbers of flu deaths to COVID deaths, or recategorizing heart attacks to COVID, or dementia to COVID, or car accidents to COVID. That's what we saw. We did see also hospital and nursing home protocols in various discrete locations killing people. We saw people dying of that. But nowhere was there evidence of any kind of biological weapon of any kind. And so this is a very dangerous narrative because it perpetuates the idea that viruses are real and dangerous.
Good evening, and welcome to another special episode of VSRF Live. I'm your host, Livio Sanchez, and as always, I'm thrilled to be here with all of you tonight. Joining us this evening is Dr. Andrew Kaufman. Dr. Kaufman is a self-proclaimed recovering physician. He has transitioned from a career in mainstream medicine to becoming a prominent critic of the allopathic medicine paradigm. Tonight, we will have a colorful discussion on germ versus terrain theory and whether or not viruses exist and are the cause of illness. But before we get started, I want to bring on our very own Nurse Angela. Welcome, Nurse Angela. How are you this evening?
I'm great, Livio. How are you doing?
I'm doing well. We talked earlier, and you're here because you have an update on a very special person that you've been really instrumental in helping get care for. Can you give us an update on Cody?
Yeah, so unfortunately, I got a phone call last night from Cody's mom, Heather, that Cody was not doing well. And for those of you out there who don't know who Cody is, he was 21 years old when he got vaccinated. He suffered an immediate reaction. He had blood clots. He was coughing up blood. He had cardiac damage. He's been really fighting for his life ever since, and it's been a miracle that we still have him here with us. He's a really, really tough kid and just super, super sweet. And this morning, I got the message that he was admitted to the hospital, and they are really concerned with a spinal stroke and blood clots on his spine. He's really, really in hard shape. And so we would like to donate any of the proceeds that we raise tonight for Cody to help out this special family. We're going to put the link in the chat for his Give, Send, Go. It's just really tragic. I remember being at Steve's house late fall of last year, and I was with Bobby Kennedy, and I FaceTimed Cody. He was in the hospital. He had blood clots in his groin and in his arms, his legs. And Bobby Kennedy said to Cody, don't give up, hang in there, just keep on going. And we're rooting for you, and we'll always fight for you. And Cody is doing just that. I mean, he is the toughest kid I know. He's just fighting and fighting. And, one of his goals is to recover and get better, even though he has a fatal condition now called antiphospholipid syndrome, which actually my mom developed that as well after her booster that caused blood clots in my mom's eyes. But he has been doing exactly that. He just wants to get better so that he can be an advocate. And he's really, really dear and special. So if any of you are able to donate, we're going to put the link in the chat. And if you donate to our organization, we would prefer it to go directly to the Give, Send, Go. But if we get any donations in tonight, we are going to donate 100% of that directly to Cody and the family, because they are, they're taking care of him 24/7. He's not able to walk unassisted, and he's in excruciating pain. But I did see a picture of him today, and even with, under the circumstances, the warmth in his heart is overwhelming to me. So help out if you can. If you can't, please pray for him, spread the word, share his Give, Send, Go, and we will continue to help take care of these people.
He's such an incredibly resilient young man. His will to live, I've never seen anything like that before.
Yeah, I don't know if I could do it personally with what he's been through. I mean, his mom has actually literally driven him in the back of pickup trucks because they couldn't get him into the car while he's coughing up blood and unable to breathe and white as a ghost. And we've almost lost this kid so many times, and he's fighting right now as we speak. So just lots of love to you, Cody, lots of love to your family, and we will help you the best that we can. And thank you for all of our viewers out there who, any little bit helps. We'll probably have thousands of people watching. So even just a small little amount goes a long way.
Yeah. And remember, everything for tonight's show will be going to help Cody and his family with their medical situation, their medical debt.
Yes.
Well, thank you. And at the end of the show, I want to remind everyone we were together at Reawaken America in Detroit last weekend, and I'm going to share a snippet of your speech at the very end of the show tonight. So we'll
Awesome.
see you again, and maybe we'll see you during Q&A.
Yes. Thank you. Thank you. Have a great show.
Thank
Can't
you,
wait to
Angela.
hear.
And now I'm going to just race into Steve Kirsch. Let me bring on the founder of VSRF. Welcome to VSRF Live. Steve, how are you this evening?
I'm good, Livio.
I am looking forward to this conversation. I was talking to Dr. Kaufman backstage before you came on, and I already learned quite a bit about my own history. So I'm really looking forward to this.
As am I.
Well, let me just bring him on right now without too much jabber. Hello, Dr. Andrew Kaufman. Welcome to VSRF Live.
Oh, hello again, Livio, and nice to meet you, Steve. Thanks for having me on.
Sure. Thanks for being on.
I'm going to quickly read your bio so that people that don't know about you get a greater understanding, and then I'll leave you and Steve at it. So here we go. Dr. Andrew Kaufman is a forensic psychiatrist and molecular biologist. He became more and more skeptical of the allopathic paradigm until he finally blew germ theory wide open and became a COVID whistleblower. Since then, he has been speaking and teaching the truth about medicine, viruses, natural healing, and other important subjects such as economics, law, education, and science. Dr. Kaufman received his Bachelor's of Science in Biology at Steve's alma mater, MIT, his MD from the University of South Carolina, and did his psychiatric residency at Duke University. Dr. Kaufman has served on medical school faculty, has been a leader in academics and professional organizations, and has conducted and published original research. He patented a medical device and ran a startup company. He has also testified as an expert witness in local, state, and federal court. Quite an impressive resume, Dr. Kaufman. Thank you for being here. And I want to remind our audience, put your questions, all caps questions, in the chat, and we will have a Q&A later, ask questions for Dr. Kaufman and for Steve. Take it away, gentlemen.
All right. Well, first of all, just for time's sake, what's the, Dr. Kaufman, I think you need to leave in about an hour and a half from now. Is that right? So we should make sure that we have plenty of room for questions from the audience?
Yeah, absolutely. That's a good time frame. I'm on the East Coast, so it's a little bit later.
Okay. So I'm going to try to target
Sounds good.
us believe that there shouldn't be a mask, any kind of mask regulations, that masks don't work, that lockdowns were ineffective, that we shouldn't have vaccine mandates, that science was not being done properly, and that there was a lack of autopsies that should have been done when people died from what they claimed is the virus or the vaccine. Is that,
all
is that
those
fair?
are good points. All good
Okay.
points, Steve.
Good. And I think we also agree on how science works, that the hypothesis that better explains the data should win at the end of the day.
Well, it
Would you
depends
agree with
on
that?
how the data is acquired, Steve, because science, we have to stick to the scientific method and its attributes and what it can be applied towards, and separate that from other things purporting to be science, which are really observation or simulation, things like epidemiology, like computer modeling, in vitro studies, things like that.
Okay, but hard data, it can be collected in a variety of different ways. That if you have the proper hypothesis, that you should be able to explain the hard data versus the model data or the artificially created data, right?
Well, like I'm saying again, Steve, the data has to be generated through a scientific experiment in order to generate a cause and effect relationship. You can have data that is worthless. Like, for example, when I was at Duke, they made a big announcement while I was in my training that they had found definitive evidence that serotonin receptors are involved in depression. And what happened is they had a large data set that was from nursing homes across the country, and they found an anomaly that suggested to them that there was a genetic finding, like of a gene for depression. And they were really excited. There was like a cocktail party, like impromptu in the department. It was like people were celebrating like mad. And then 2 weeks later, I noticed that there was no further talk about it. And what had happened is several other groups with similar data sets found out it was just a statistical anomaly because they were data mining, and they essentially encountered an anomaly and then misinterpreted it as an actual finding. So that's because all they had was a big set of data. It wasn't data that was from an experiment designed to test a hypothesis of cause and effect. It was just kind of data that was for another purpose, for another experiment, that they were just put into a big number crunching, software, and essentially ran simulations and then came up with false conclusions.
Okay, so I'm sure we could spend another hour on that topic, but in the interest of moving on, let's just say I don't completely agree with what you're saying. I think that data is data, and you can what science should do is interpret that data and assess whether or not that data is going to be reliable or not. And
Well,
that's
now,
part
if
of
we
this.
can't,
That's
if
part
we-
of science.
If we can't stick to the scientific method, where there's an independent variable, there's a control, there's a dependent variable, that's the only way you can essentially ascertain scientifically any cause and effect relationships.
Okay, well, like I said, I think we're going to have to agree to disagree on that, but
Well, no,
let's
this
move
is
on.
not, I mean, this is, the scientific method is well published and agreed upon throughout the world of science. So if you disagree with that, I don't know what we could talk about.
Okay. So let me give you an example. I claim that if you take a golf club and you hit a golf ball, that the club is actually causing the golf ball to move, and it's the contact with the club that causes the golf ball to move, and it's not the air of the golf club that causes the ball to move ahead of the club hitting the ball.
So
Okay,
you could
but I
design
don't need to go. Do I need to do a? I don't believe that I need to do a randomized control trial in order to figure stuff like that out. Or if I jump out of a plane with a parachute and the parachute doesn't work, will I die or not? We don't need to do a randomized double-blind trial on jumping out with or without a parachute.
Well, two things. One, a randomized double-blind trial is not necessarily a scientific experiment because it's not designed to highlight or prove a cause and effect relationship in nature. It's done for pharmaceuticals, which are man-made. But the other thing is you could actually, I mean, now, of course, I agree, you don't need to necessarily do an experiment to see if the club is moving the ball, but you could do an experiment, right, where you could swing just air, and you could swing the club, and the club would be the independent variable, and the dependent variable would be the motion of the ball. And the control would be swinging air instead of swinging the club. So when you're talking about invisible things, you do need to very carefully design and perform these experiments because it's not so obvious as observing someone swinging a golf club.
Okay, so you're saying that all retrospective observational trials are completely useless because you couldn't control anything in the environment that you're observing. You had to take the data that you were given. And you didn't control for that. You didn't. You control for it after the fact, but you tried to control for it after the fact. But you're saying that observational studies are junk. Is that?
No, I'm not saying they're junk. I'm just saying they're not science because you can't make any cause and effect conclusion out of. Well, let me just finish.
Yeah.
So observational data like epidemiology is only useful to generate hypotheses. You can't conclude anything. If you can conclude things from that, then you're basically saying that causation equals association.
Okay, well, like I said, we could spend a lot of time going down this particular topic, but I really want to focus on what you're about here with the whether viruses exist or not. So I think we just need to table this because otherwise we're just not going to get anywhere in this call.
Well, this is the framework for this discussion, Steve, because we're going to talk about a scientific claim of cause and effect of a natural phenomenon. And so you can only get to the answer of that with using the scientific method. You can't get to that answer with epidemiological data. And any epidemiologist will also admit that.
Okay. Well, let's just move on, and maybe we can go back to this point if we need to. But would you agree with me that science, if you should have repeatable results, they should be explainable by your theory? For example, gravity has been replicated in a number of different locations all over the world. Things don't fall to Earth and accelerate any faster than gravity, or any less fast than gravity. It all accelerates at the rate of gravity, and people have observed that all over the world with must be millions of observations. Would you accept that gravity is a scientific fact?
Well, you can observe gravity as a fact of nature, but nobody knows what causes it.
No, I didn't ask that. I was just saying that if I release an object, it's going to go and fall and accelerate at the gravitational force is going to accelerate that object when I drop
Well,
it.
I mean, of course, it has predictable behavior. There are equations you can apply and predict it, with some degree of error, but fairly accurately.
Okay.
No, I don't debate that, but
Great.
once again, that's not a
Okay,
cause
good.
and effect relationship.
Okay. Well, I'm gonna move on to the next point, which is that we would agree that all scientists should be open to public challenge, right? And that if you say
Well,
something,
I mean,
that you should be open to being challenged by your peers on what you say. Would you agree with that?
the main principle of science is falsifiability. So it's necessary for science, not just permissible.
So you agree with me that people should be open, whether people agree with you or disagree with you, that you should be able to challenge them publicly and they should be able to challenge you publicly.
Well, I mean, if you participate in a public discourse, you're always open to challenge, but I'm not going to say what people should and shouldn't do. But I will say that if you're serious about science, then a scientific hypothesis or theory has to be falsifiable. That's one of the standard principles that defines it as scientific.
Great. And the goal is to seek truth, right?
Well, that's my goal.
That's
You
my
know, I mean, I would hope a lot of people that do science, their goal is to be rich and famous, maybe, or some other purpose than finding the truth.
Right. Okay. So, and we also agree that there are scientific procedures such as the PCR test that have design centers for the tests, and they're designed to be used in a certain way on a certain thing. So if you use them improperly, you can get results that are nonsensical. So let me give you an example. We would agree that a PCR, that's a lateral flow assay test that is designed to detect whether a human being is infected with SARS-CoV-2 may in fact have a false positive if it was given a papaya or something else, but that doesn't necessarily mean that the test is invalid. It means that you did not use the test in accordance with the manufacturer's recommendations because the test was designed with certain limitations in terms of how it should be used.
Well, I don't think really we should be discussing tests because tests apply, to something that's already established. But what I would say is that a test, you can't presume a test is valid. You have to actually validate the test first. So if you were to validate a test, for example, that was going to diagnose someone with an illness caused by a virus per se, well, then you'd have to do a validation study where you have a population of people with that viral illness and other people that either have an illness that's not viral and people who are healthy probably as well. And then you'd have to be able to find the virus directly in the people who are sick and look for it in all the other people to make sure you don't find it, and then do the test that you're proposing in all those people. And then that's how you actually calculate those error rates, which, like false positive, false negative, and, any other types of errors from that. But for all the COVID tests, they didn't have to be validated because of emergency use authorizations. So those things were never done, those studies.
Right, but that wasn't my question. You didn't answer my question.
I thought you were saying we agree on that that tests could be misleading if the procedure is not followed.
Correct. So if you go and you use a PCR or a lateral flow assay test to assess a human being, that if it's used to, that if you take a lemon and squeeze it on the lateral flow assay test and you get a bar on it, that's meaningless because the test was not designed to be used in that way. I'm asking about
I would say if the
test
test
abuse.
isn't-
I'm not asking you whether the PCR test or sorry, the lateral flow assay test works. I'm not asking you whether it's valid or anything else. I'm just saying that would you agree with me that if a test is used in a way that it was not designed to be used, such as trying to use a PCR test on or a lateral flow assay test on squeezing a lemon, that it's perfectly reasonable that you could get nonsensical results. Would you agree with that?
Well, Steve, I'm not really sure why you're asking. This is kind of a strange question, but let's say you had a pregnancy test and instead of peeing on it, you licked it. I wouldn't expect you to get an accurate result if
Right.
you did that. That's
Good.
correct.
Right.
But a pregnancy test has already been validated in a sample population.
No, I'm just saying that if you looked on the present pregnancy test, you should not pay attention to the results. I think we agree on that, right?
Yeah, I mean, I don't know, maybe
Good. Good. I'm just trying to.
could be right, but I wouldn't advise it. Yeah.
Okay. All right. Great. And then I want to mention before we get started here into the specific questions that this is not my area of expertise, genomic sequencing. I admit up front that I could probably ask questions that may be nonsensical or make mistakes just because I do not claim to be an expert in gene sequencing, and you know that.
Well, I'm not an expert in gene sequencing either.
Okay.
Actually.
Great. Well, great. Great. So let's start off with some real simple softball questions just to get this going here. So your name is Andrew Russell Kaufman, MD. Is that correct? That's a yes or no?
Well, I wouldn't say MD is part of my name. It's
Okay, but your middle name is Russell. Just to make sure that we have
I can tell you for sure that Andrew Russell are my name, but it would only be hearsay that Kaufman's my last name. I have no direct firsthand knowledge, but I go by it.
Okay. And have you published papers in genomics? Have you published any papers about your theories about whether the COVID or whether the SARS-CoV-2 virus exists? Are there any papers either on a preprint server? Are there any published papers or even unpublished papers that people would want to take a look at?
Well, there's a paper I wrote with Tom Cowan on my website, but there's a very, very low likelihood that any journal would publish any paper that I would write on this topic, because if you look at some of the other attempts at publication, even in much, much less controversial areas, like the Dutch group who actually did a randomized controlled trial of masks during COVID, they had to basically violate Statistics 101 to get their paper published after 3 rejections. So that would be kind of a fool's errand, in my opinion. So I never pursued that.
Right. No, I get that. But you have a paper that's available on your site that people can read that explains how all this works and what your theories are.
Well, it only specifically talks about isolation experiments and electron microscopy. But yes, it gives the basic ideas. But I have lots of published lectures, like slideshow type lectures that explain all the arguments, and even a documentary film.
Okay, so people can go to your website, and you want to give us the URL for that?
Yeah, it's just andrewkaufmanmd.com.
Great. Awesome. Easy to remember. Okay, so let's get into the key question here, which is, I believe that I've seen the Del Bigtree interview. I saw the excerpt that Debunked the Funk posted to the web, where you were answering some questions. So I tried to do my homework, and it appears that you claim that, and so you can correct me if I'm wrong, but I'm trying to summarize that it appears that your opinion is that that SARS-CoV-2, the virus, isn't in fact a virus, that it's an exosome causing this, that SARS-CoV-2 doesn't exist. And so I believe I got that part right, but I wanted to make sure that we're crystal clear on the second question, which is, do you believe that any viruses exist? Do you believe that the measles virus exists? Do you believe that smallpox virus exists? Do you believe the polio virus or poliomyelitis virus exists? Or do you think that there is no such thing as a virus? So let's just start with SARS-CoV-2 just to make sure that I got that right. And then if you can answer clearly the second question as to whether you think any virus exists, and if so, then which ones?
Yeah, sure. So you're partially correct. You're in the ballpark about SARS-CoV-2.
I try. Okay.
Basically what I've said is that the claims put forth that this virus, exists and is causing disease are baseless. Like if you actually look at the published studies which assert those alleged facts, they did not meet the bar for proving anything or demonstrating everything. And we can, of course, talk about that in detail. And, in my journey of discovering this, of course, I looked at many other alleged pathological viruses, right? Those are alleged viruses that are said to cause disease, like you mentioned a number of them. And I haven't personally looked into every single virus that's in, a microbiology textbook. But I've looked into all the ones you mentioned and several more, and then some of my colleagues who also have examined the same material and have similar conclusions, they've looked at the rest. So there's a large body of work out there, some of it in written form, in papers and blogs, and a lot of it in video form about all of those different viruses. So in terms of viruses that are alleged to cause disease in humans, I haven't found any prima facie evidence that any of them actually exist in the real world. There are other nanoscale particles that are real, that live within microorganisms, like, for example, bacteriophages, which are often compared to viruses, but they're able to actually demonstrate their existence in reality.
Okay, so you agree that bacteriophages do exist and they do infect bacteria?
Well, their actual role in the bacterial culture is not nearly as clear-cut, but they definitely exist because you can find lots of studies where they're purified and the only thing you see on a microscope image is identical, very uniquely shaped particles that,
Okay.
it's obvious that it's one thing you're looking at.
Okay, but as far as you're concerned, you do not believe that viruses exist, or you have not seen any conclusive proof that viruses exist, even though bacteriophages exist.
Correct. The
Okay.
ones that are alleged to cause disease.
Okay, so that's interesting because if you were to look up bacteriophages, you would find that they're considered to be viruses.
Yes, well, that's why I'm kind of specifying what I've looked at, because you could call them that, but what the functional definition of virus hasn't been fully established for bacteriophages either. But
Okay,
they
so-
definitely can demonstrate that they're real.
Okay, so we're talking about nomenclature that in bacteriophages exist. They do infect bacteria. But
No. I'm saying that there's no evidence that they infect bacteria. They're present in bacterial cultures under very stressful conditions in a pure bacterial culture, which is not a natural
okay,
setting.
all right.
But there's no experiments that show that they cause an infection to bacteria.
Okay. All right. Okay. Then we'll get into this more later. But I just want to kind of establish your position. So essentially, you're saying that from your point of view, you're not convinced that any virus exists. That you would probably say that it's possible that the SARS-CoV-2 virus could exist. It's possible that the measles virus could exist. But from your perspective, you haven't been scientifically convinced that these viruses do exist. Is that Would that be fair characterization?
Yeah, there's just not sufficient evidence to demonstrate their existence.
Great. Okay. I'm going to start off with a bunch of short questions, softball questions. Hopefully I can get a quick answer on these because they should be yes or no questions. I saw this video that was posted by Debunk the Funk 3 years ago on YouTube, where Tom asked you a question at one of your seminars, and you said that you would follow up and you would examine the paper and get back to him. And he also
You're
suggested.
talking about when someone sabotaged one of my webinars and got access to their camera and stuff and then made a video trying to make me look bad and call me names and say I didn't go to medical school. You're talking about?
I'm talking. Yeah, I think we're talking about the same person,
Yes,
and I'm not
yes.
sure that I can reveal his last name, but he was referred to by Debunk the Funk as Tom, and he basically said, "Here's the paper," and you said on video that you would review that paper and that you would discuss it with him. And I'm just inquiring as to whether that discussion took place, and if not, why not? And also that he suggested that you take a course in I think genomic sequencing. And I was wondering if you took him up on that offer as well. Simple yes or no question.
Now, Steve, this is what you call a logic fallacy here. You're trying to defame my character by saying I didn't, have a discussion with the guy that I said I would.
I'm just trying to ascertain. I'm just trying to ascertain whether you did. I can't be defaming you because I'm not accusing you of not having the discussion.
I want to only talk about the scientific issues, not about some random guy who tries to insult people on camera.
Well, this is not a random guy, and he presented scientific evidence to you, which you said that you would have to look further into the details of the methodology.
I made a video interpreting the paper that he gave, so you can find that on my platform.
Yes, I'm aware of that. Okay, but you never engaged with him on.
I'm not going to engage with people who insult me.
Okay,
on video,
right.
on camera. I
Okay.
mean, why would I do that?
I didn't think
That's
that he was
not
insulting you.
That's not. Yeah, he called me names and said that I didn't graduate medical school or something, and put up.
Okay.
Freeze frames of my face in an awkward position. Kind of
Okay.
teenage
Okay.
stuff like
All
that.
right. Okay. So let's move on to the second question. Is it possible to get to passage 4 stock if there's no replication?
Wait, say that again.
Is it possible to get to passage 4 stock if there's no replication?
Well, a passage is just taking some fluid from one cell culture and adding it to another cell culture. So what do you, all you need is a pipette and cell cultures to do that. Is that what you're asking?
Well, you understand what I mean by passage 4 stock, right?
Are you talking about?
It was in the original. It was in the okay. There was an original sequencing of the SARS-CoV-2 genome. It's in that paper. This is a direct quote from the paper, which I'm sure you're familiar with because that's basic literature that everybody would be familiar with who looks at SARS-CoV-2.
Are you talking about the paper I sent to your staff, the Fan Wu paper?
I don't think it was.
The Fan Wu paper is the first published genome of SARS-CoV-2.
No. The first one in the United States.
I'm not familiar with that.
Let me see if I can look it up real quick.
Only the first genome matters because they used templates for all subsequent genome sequencing. So
Right. Okay. So let me.
that's why
let me
I
s-
submitted the Fan Wu paper to your staff. Did you have a chance to read the papers I submitted?
No, because they didn't provide it to me. So
I
this
gave it
is
several days ago, Steve. Why didn't you have a chance to read it?
because they never. I'm telling you, they didn't provide it to me.
I see. Well, why did
Let's see.
Why did
Okay,
they ask
so-
for it then?
Okay, so the paper that I'm talking about is titled "Isolation and Characterization of SARS-CoV-2 from the First U.S. COVID-19 Patient."
Okay. I mean,
Okay.
I read that paper, but I read it, a couple of years ago.
Okay, but let me let me read the, let's see. So, I'm looking at the paper right now. We subsequently generated a fourth passage stock of SARS-CoV-2 on Vero E6 cells, another fetal rhesus monkey kidney cell line. Okay, viral RNA from SARS-CoV-2 passage 4 stock was sequenced and confirmed to have no nucleotide mutations compared to the original reference sequence. And then they give the GenBank accession number in that paper. So how is it possible, Dr. Kaufman, to have a fourth passage if there's no replication?
Well, what is the starting materials of that experiment, Steve? Did they have a virus and put it in there? Is there a virus in that experiment?
You said you read the paper. This is
No, I said I read that paper 2 years ago. You think I remember the details, but I know how all these papers are done similarly. What was the starting material?
It doesn't matter. This is a generic question. I'm simply asking you. I'm simply
Steve,
asking.
Steve, you're saying the
Hang on.
experiment-
Just let me. You're interrupting me. Is it possible to get to passage 4 stock if there is no replication? This is an easy question that when I ask any of my experts who do this stuff for a living, there is no question as to what the answer to that question is. And so it troubles me that you're asking questions like, how is this prepared? This is a simple, cut and dry, objective question. Is it possible to get to passage 4 stock if there's no replication? Yes or no?
Steve, I can't answer that question because I don't know what you mean, is it possible? Like I said, passage just means you pipette some fluid from one culture and put it in a subsequent culture. So you can do that a million times, right? As long as you have enough pipettes and enough cell cultures. You're interrupting me now.
Okay. I was going to try to answer your question.
In order to understand what this experiment, because you made that question completely out of context. What is the experiment like? What are the procedures? How many samples do they have? Who are they from? Is it a one sample from one patient? Did they purify a virus out of it? Did they prove a virus is in it first before they started the experiment? How do you know there's a virus in the experiment?
So what they did is a patient was, so this is, I'm reading directly from the paper. A patient was identified with confirmed COVID-19 in Washington State on January 22nd,
We're talking
2020.
about a paper that proves that COVID exists. How can you start with already knowing that they have COVID?
Because we're talking about COVID-19, which is the disease, not SARS-CoV-2, which is the virus.
Gotcha. How do you diagnose COVID, though?
Well, let's see. Okay, so what they did is they used a cycle threshold of 18 to 20, both in the throat swabs and nasal swabs.
Okay, so they did a PCR test, right?
They did a PCR test.
Okay, so
correct.
Now how do you know that the PCR tests can diagnose COVID?
They didn't say that. They just said that it was confirmed that the PCR test said it was COVID, and that they did.
That's the whole.
But
the whole
they're
thing
doing
is based on that patient has COVID, right?
Okay.
And they're saying they have COVID because they have a genetic test, right? That the genetic test is supposedly testing genetic sequences from a virus. But this experiment is to prove that there's a virus. So they assume there's a virus. They have a test for it. That without ever finding it, and then they use the test results to prove that it exists. This is circular reasoning. Where's the prima facie evidence that there's a virus in the first place? You can't have a PCR test without that because the genetic sequences allegedly belong to that virus. How do you know they belong to it if you haven't discovered the virus first?
Well, but they did discover the virus first in an earlier paper out of China. And so these
Are you talking about
PCR
the one
tests
that I
were.
submitted to your staff, the Fan Wu paper? That
Oh,
is the first genome?
it probably was, but I don't know what the origin of the PCR test was in this paper, and maybe if I had time to read it more carefully. But my point is this: that that everybody I've talked to says it's not possible to get to passage 4 stock if there's no replication. So you're claiming? Are you saying that?
What does that mean? How is it not?
Question.
What's not possible about it? You mean that you won't have a positive PCR test in the
No,
fourth pass?
no, that you can't get to the next stage. In order to get to passage 4, it means that there are basically 4 replications where you measured that the thing that you're looking at has replicated.
When you say replication, what do you mean?
Well, in order to go and do genomic sequencing, you usually start off by taking a sample.
About cultures, aren't you? Or are you talking about
Yes, cell
sequencing?
cultures. Yes, no, cell cultures.
Okay.
Before the sequence. You don't just take the swab and then try to genetic sequence that. You would go and try
No,
to.
you're right. You got to take genetic material from an actual organism to sequence it, so you know what you're sequencing.
Right, but then you go and you put it in these Vero cells in order to replicate it, so that you have lots of copies of it, and then you go and run the sequence on it.
Why would you do that? You can amplify it with PCR. You only need 1 copy and you can make an infinite amount with PCR.
You
Why
could.
would you need to culture it?
Because you want to culture the entire genome. Does that make sense?
You can't culture a genome, Steve. You can only culture organisms.
Yes, but you're culturing the virus that you believe exists. And what happens is that they do this and they find that the virus replicates.
You're saying that you prove that you're culturing the virus by detecting these sequences that you can't prove came from the virus?
What you can do.
How do you know the virus is in that experiment?
You don't necessarily know that, but you're replicating something. Something's replicating. Do you think?
How do you know?
Do you think you can get to passage 4 stock if nothing is replicating?
Like, what do you mean by replicating? How do you tell if something's replicating?
Well,
like if
because you can
replicate,
because if you have twice as much as something, and you then go through another process of replication, and you have double that amount, then you believe it's clear that something is replicating.
You have twice as many viral particles?
You have twice as many particles.
Okay, where are the pictures of the particles going from half as many to twice as many?
I'm just asking you. The point is that when you go, these are pretty. I didn't expect to be stuck here.
That all your results that you're talking about are based on PCR testing, with looking at. Well, then what?
Passage 4 is not about a PCR testing. It's putting these samples into Vero cells and having them replicate in the Vero cells.
How do you detect their replication?
Like I said, I'm not an expert on this, but I've talked to people and they don't think this is a problem. Have you ever done this stuff before, Dr. Kaufman, yourself?
Done which stuff? Have I worked in a laboratory with culture, cell cultures?
Have you ever taken a swab of somebody who has supposedly, hypothetically, a virus? Have you ever taken a nasal swab, and have you ever placed that into a Vero cell and then replicated that and then done a whole genome sequence on it?
Do you know what a Vero cell is?
Yes, it's a monkey kidney cell.
So let me ask you, if this virus grows in the lungs, why do they need to use monkey kidney cells? That are genetically modified, by the way.
Surely, Dr. Kaufman, I can't believe you're asking me that question. You're telling me that you don't know the answer to it? I'll tell you the answer to it, but
I know the answer to it, but I think we have different answers.
Well, okay, well, I can tell you the answer to it as far as I know. And again, I'm not an expert on this, but the reason they use these cells is because these cells are basically, these immortal cells that are easy to grow stuff in. And so it's simply a scientific container to allow a replication of a virus that comes from a human or some other places, and it happens to be a nice medium for growing things. It's like dirt, effectively. You know, with dirt for plants, or you take a plant, you put it into dirt, the plant grows. The same thing can be true for putting these foreign cells, this virus, into vero cells, so that you can go and replicate the virus.
So, Steve, this is a good point, because the paper number 2, if Livia wants to pop it on screen at some point, talks about this, that SARS-CoV-2, even if you think it's real, it's even mainstream people say it doesn't affect the kidneys. So how could you culture it in kidney cells? But this article shows how they have kidney biopsies from pre-COVID and during COVID, but with all negative, unvalidated COVID tests, and they found particles on electron microscopy that look identical to what they say coronaviruses look like, the same size with the spikes. And they basically say that you can't identify it from these images, and that this problem has been known about since the 70s. This is what it says in the paper. And even there's another paper that the CDC published about this issue, that by looking at these microscopic images, it doesn't tell you what you're looking at, that these things could be confused for all types of subcellular particles. And that's what the exosome discussion came from, because exosomes are just one type of many subcellular particles that are found in the body, and they're much more common in pathological conditions like disease and injuries, that they can't be distinguished morphologically by sight, or by shape, by appearance on microscopy from each other. Okay, there needs to be some other way of identification. Just like in the experiments you're talking about, there was no direct visualization of viral particles that are being replicated.
There doesn't need to be. We have whole genome sequencing.
Well, if you don't detect the actual thing you're looking at and use a proxy variable or a covariate, it needs to be validated. So if you're
it is validated. The whole genome sequence, Dr. Kaufman, do you know how many countries have validated the whole genome sequence of the SARS-CoV-2 virus?
They haven't validated it. What they've done is they've run a template and not gotten the same results, which is why there are millions and millions of variants.
Yes, but they're very similar, aren't they?
I don't know. We're very similar to chimpanzees, but we're not the same.
But the size of the genome for a chimpanzee is quite a bit different than the size of the genome for SARS-CoV-2. Do you know what the size of the genome for SARS-CoV-2 is?
There is no genome for CoV-2 because SARS-CoV-2 hasn't actually been shown to exist. Let me call it the gene sequence. The sequence of nucleotides for SARS-CoV-2. Do you know how long the sequence is? Steve, let's look at the Fan Wu paper, because
that's a simple question, Dr. Kaufman. You don't know how long it is?
There is no valid genome. I know how long they say it is, but it's not valid.
How long do they say it is?
It's roughly 30,000 base pairs.
Is it more than 30,000 or less than 30,000?
Well, we can look at this paper. It has the exact number, because this is the paper that...
No. But their sequence...
It's not that one, Livio. It's the fourth one, number 4.
There are sequences, Dr. Kaufman, from over 100 countries in the world. And Steve, it's amazing. It's amazing that 100 countries all detect sequences of virtually the same length, with virtually the same structure, with minor differences here and there in the sequence.
So it's a logic fallacy to say that just because something has been done by a lot of people that it means something. Now,
Really? Okay.
Yes,
So how do you
that's a lot.
how do you explain that over 100 countries get the same results for whole genome sequencing? You know how it works, right?
Steve, I already said, because you don't know what the procedure they're doing. They are using a template. A substantial portion of the genome that they're looking for is contained in primers that they're using, and they're templating to this genome, which is, I'm trying to explain. If we look at the procedure, so all of those other ones, they're templated and validated against this original genome, which was the one first
Yeah.
recorded in GenBank.
Yep.
And this one, if we can go through the procedures, you'll see that it's nothing more than a computer simulation. It's not an actual experiment where they took an organism and took out the genetic material and sequenced it. You know, that's how they got the genome of humans and cats and dogs and chimpanzees and bacteria as well. But here they do something very different, which is a simulation. So if I can just get the paper up on my screen here for a second.
See, here's what I don't. You understand how whole genome sequencing works, right?
Well, that's not what's done for
No,
virus.
it's a yes or no question, Dr. Kaufman. It's a yes or no question. Do you understand
You
how
have
whole
to
genome
define-
sequence is done?
You have to define the term.
Whole
Because
genome
there's many-
sequencing? You have to define the term? Are you serious?
Listen, that's not a term I've seen in the literature, so why don't you just tell me what it is?
Wow, I'm flabbergasted. Do you know what hybrid sequencing is?
Hybrid sequencing. No, I'm not familiar with hybrid sequencing.
Do you know what? Have you heard of?
Steve, are you trying to avoid talking about this paper, which is actually what they did, or instead
No,
do you want to test
no,
my
I'm
knowledge
really not trying to look. If
vocabulary?
Dr. Kaufman, if we had the next 8 hours, I'm happy to go down into any rabbit hole that you want to go down at. I'm
This
trying
isn't
to
a rabbit hole.
focus.
This is the scientific paper that establishes the genome that you're saying is the evidence
No,
for.
but see, that's not the important thing, Dr. Kaufman.
It's not.
No, it's not.
It's not.
It's
Okay,
not.
what
No,
is?
no. You should. Do you know what have you heard of Oxford nanopore?
I think we should talk about contagion.
Yes or no? Do you know? Are you familiar with the Oxford nanopore system?
This line of questioning with you naming brand names of molecular biology test kits or, different procedures is not going to allow us to learn anything.
Yes, it is. Yes, it is. That's the reason I'm bringing it up. It's a simple yes or no question. If you would answer the question rather than try to evade the question, then I will explain to you why this is relevant. Do you know what the Oxford nanopore system is? Yes or no?
No, I don't know what it is.
Okay. All right. So,
Do you know what metagenomics is, Steve? Do you know what deep metagenomics is?
I'm familiar. Like I said, I'm not an expert. But you
deep
claim to be an expert in this,
metagenomics
Dr. Kaufman. You claim to be an expert.
Well, I don't claim to be an expert.
But I'm asking you some basic question. Like, you don't understand, Dr. Kaufman. Here's the point. Let me just summarize the point here because we only have 7 minutes left. Over 100 countries throughout the world have done whole genome sequencing. If you go and you look at, for example, ATCC, when they do their validation, they use the Illumina system to do their sequencing, but they also combine it with the Oxford nanopore reads. Now, Illumina reads about 300 or so bases typically, sometimes more, sometimes less. The Oxford nanopore system is much greater, that they can read sizes that are huge in comparison, much larger than the SARS-CoV-2 sequence. And so you don't have any of this, you can't do any of this hand waving to say that this is mathematically generated from primers because they use random primers when they do whole genome sequencing. They don't go and go to Wuhan and get the primer sequence and just try to replicate it from that. They're basically replicating the results from scratch. They take the material, they're not looking at any reference papers, and they're able to come up with the exact same sequence. And they're able to do that throughout the world. And you seem to be unable to understand that, and you don't even understand the whole genome sequencing technology, and you've been misleading people. I believe that you've been misleading people because you've been saying that they assemble these things in a mathematical algorithm, and you're not even talking about the hybrid sequencing that they're doing that completely eliminates that possibility. And you've been telling people that, oh, these sequences that you can get anything that you want out of them. I've heard you say that, and that's just simply not true. And you don't seem to be familiar with how it's done. You've never done it before.
All right, may I respond now?
Yeah, go ahead.
So, Steve, I am familiar with exactly how it's done because I actually read all the scientific papers where they establish these things, and I tried to send you these papers, but I'm not sure if you actually read them because you need to go step by step. Now, the specific technology that's used to actually take a strand of genetic material and sequence it, there are many ways that it can be done, and I never said that any of those ways in and of itself were a problem. The problem is that what they do is they sequence genetic material that they don't know what organism it's from. So in the original genome sequence, the Fan Wu paper, okay, they only had one single patient, first of all. And they took bronchoalveolar lavage fluid, which is from the lungs, so lung fluid from that patient, and they extracted all of the RNA in that patient's lung fluid. Now you can imagine that there are hundreds of different organisms in that lung fluid and also in whatever was in the last breath that they took in, right? If we just swab the air, we can find DNA and genetic material. So they then, made a cDNA library and then used next generation sequencing, which is attaching, small oligonucleotide strands to beads, and they're all sequenced in parallel, and it really speeds up the process, and it's a good technology. But when you start sequencing with material of unknown origin, unknown provenance, then what you get out of it are sequences of unknown origin and unknown provenance. And what they did is they got over 56 million sequences, they call them contigs, out of that next generation sequencing data. And then they use computers to simulate and modify the sequences. So for example, one thing they did is they removed sequences that they said were ribosomal RNA. They also say that they removed some human sequences, but they didn't actually publish their exact procedure. And humans we don't all have the same sequence. So it's not a trivial matter to identify a sequence as innately, human versus from something else. Even bacterial, there are short, nucleotide sequences that we overlap even with lower organisms. So basically, out of these 56 million strands, they put them into not one, but two different software programs. And these software programs use different algorithms to try to match the smaller strands into bigger ones by having various overlaps in their sequence. And they, of course, constructed many, many solutions. One software program had over 3 million solutions. One had over 1 million solutions. And from these, 5 million possible completely made up genomes where every piece of sequence RNA in them comes from an unknown source. Could it be human? Could it be a bacteria? Could it be, something else unknown, another virus, a horse that somebody was riding the other day, almost anything. And then they, out of those 5 million computer simulated sequences, they pick one. But that's not good enough. They say, for some reason, even though they say that's accurate, that they still don't know what if the ends are right. So they have to make primers to the ends of those sequences and then put it back in the original mix and do more PCR and then sequence what gets added on the end, and then report that that's what, but first they do some further processing to substitute some nucleotides to get the right amino acid codons. Then they publish it as being something real. So this is an entire computer simulation from a mixture of unknown sources of DNA. In this entire experiment, there's not once demonstrated that there's a virus.
So can you explain, these people who are doing this in various countries are doing de novo whole genome sequencing. So
Now,
how did they get the same result?
Steve, tell me what the procedure that they're doing, step by step, because I just gave you the step-by-step procedure. Now you give it to me.
You know, like I said,
I'll tell you if it's valid. I think the audience will know if it's valid. I think they know from my
but
description.
the point is that it doesn't, it's irrelevant, right?
No, it's not. How can I answer the question if I don't know what was
Because
done?
they're doing de novo whole genome sequencing.
The experiment that I just described, do you think the genome sequence at the end of it is a real genome of a real virus?
Yes.
Based on what I said?
Because
How do you know where the sequences came from?
well, because you know, in the paper it said, hey, you know, we took a swab from a patient who is exhibiting exhibiting COVID-19 symptoms. Okay,
Now,
so the origin is human.
are not distinct from pneumonia. They're the same exact symptoms. In that paper, if you read it, you'll see they just had pneumonia symptoms. So
Yeah.
how do you know they had a virus in them?
Well, because they're able to extract out a DNA sequence that they've never seen before. So, for example, you're familiar with the furin cleavage site.
So because they use computers to simulate a sequence that's
No,
known, it has to be a virus? That's what you're saying?
No, I didn't say it had to be a virus.
My question was, how do you know there's a virus in that?
Because they look at the sequence that they extract in the whole genome sequencing that's done de novo.
The experiment where they're determining the sequence, Steve, they can't determine it and know it ahead of time at the same time. That's begging the question.
But do you understand what de novo whole genome sequencing is?
Steve, forget about the term.
Wait, that's a yes or no question, Dr. Kaufman.
How do you know that sequence that was the result of the experiment came from a virus?
We'll get there. Let's just.
Question, Steve.
I asked you, do you know?
Answering this question, it's purposeless to talk about genomes at all because this is the first genome. If this genome is not accurate, then every genome subsequent to it is not accurate. So
that is not true.
Those sequences are from the virus.
That is not true. What you just said is completely false.
I'm not going to say anything else until you answer this question.
I asked you if you knew what de novo whole genome sequencing was. You're obviously not very familiar with whole genome sequencing. It appears you don't know
about deep
what
metagenomic sequencing. That's what was done for SARS-CoV-2. So how do they know the sequence is from a virus?
Because they can look at it. They can look at the sequence and determine whether it's a virus.
There's no pictures in the paper. They didn't do any microscopy.
It's called sequencing. It's called genome sequencing.
But how do they know?
You seem to be unfamiliar with this technology. You still are not answering my simple question, which is whether you know what de novo whole genome sequencing is. Yes or no?
Steve, the sequence came from lung fluid. That's a human fluid. So are you saying that it represents the human genome?
No, the human genome.
The virus
genome is filtered out, Dr. Kaufman. You should know that.
I'm going to step in
because it's filtered. Because we filter out the stuff that we know about, and what remains, we say, "Oh, well, this is interesting."
What remains is a virus. There's no other unknown organisms out there.
There probably are, but there's one that is replicated that has that you've not seen before.
How do you know it's not another unknown organism? How do you know that all of the I'm answering your question. Okay, you asked me how do I know it's not another unknown organism? Because people throughout the world have done this
millions and millions of times,
again, that's a
and they
fallacy.
get
Just
exactly the
because
same results using
they
de
don't
novo
get
whole
exactly
genome
the same
sequencing.
Okay, all right.
results.
I am
They don't
gonna
get the
I'm gonna
same
call
result.
a timeout. I'm gonna pull an audible. I'm gonna step in the middle of this as awkward as it is for me. Um, Thank you for Dr. Kaufman. I want to thank you also just for being present the way you are and handling everything and all the questions. And Steve, I want to thank you for being tenacious as you are. But I'm going to step in and go to some audience questions that I think everybody wants to hear, and proceed forward.
Okay, one question before we do that was a very simple question. I just wanted to clarify, Dr. Kaufman, do you believe that the SARS-CoV, that people who have COVID, that is being caused by exosomes, yes or no?
Well, I don't really have any beliefs about this, but no, exosomes don't cause anything. They're a result of diseased cells.
Okay, thank you.
All right, I'm gonna move on to some audience questions because I think they want to hear the answers to these, and I'm gonna start with number one from our very own nurse Angela. If HIV patients aren't dying from HIV, and this is for you, Dr. Kaufman, what are they dying from?
So, for everybody that wants to know what my hypotheses are about the cause of other different diseases, I have a healthy living live stream every Wednesday at noon on all my channels, YouTube, Instagram. That is not relevant to discussion today because that's what's called a straw man fallacy. And I'm not making any claims. I'm refuting the claim that this virus or that virus causes a disease. So in order to refute that claim, I don't have to have any alternative explanation. And there's really no scientifically valid explanation as terms of cause of most of these diseases. There may be some things. Some of these diseases, if you start looking at it, it's not even clear that the disease exists. I'm not talking about AIDS, but if you do look at AIDS, all of the AIDS-related conditions, right, like Kaposi sarcoma and pneumocystis carinii pneumonia and various other conditions, they all existed before AIDS. In fact, very shortly before the AIDS crisis started in Northern California, there were reports of Kaposi sarcoma in kidney transplant recipients because they had invented a new transplant rejection medicine, azathioprine, and it resulted in Kaposi sarcoma in some of the recipients. So it was kind of interesting that later on that was hidden, but there are many causes, potential causes of these things, and we need to do some very rigorous, well-designed, well-controlled experiments to make solid determinations.
Fair, yeah, okay, makes sense to me. And a follow-up from Nurse Angela. When one kid gets chickenpox, many other kids get it at the same time. Another.
Yeah, so that's now that, I don't know if someone actually observed that or they're just saying that other people say that. But I'm glad you brought up contagion because recently, and I did, by the way, one of the papers I submitted was a contagion study from the Spanish flu done by Rosenau from the predecessor of the CDC, the National Health Service, and it was done, with Navy soldiers, volunteers. But turns out there are over 90 actually scientific controlled studies of contagion where they're trying to use various techniques to pass an illness from one person to another. And sometimes they do animal studies as well because some common human illnesses like the flu, animals are susceptible to as well. So this set of experiments that you put up on the screen, for example, I think there's 8 total experiments where they had a variety of different body fluids, bronchial fluid, saliva, nasal secretions, even blood from patients in the hospital during Spanish flu outbreaks. And they put them in a bunch of volunteers in many, many different ways. They squirted it up their nose. They had them get really close to each other, and one exhaled and the other inhaled. And they did this on a Spanish flu ward where everyone had the Spanish flu. They even did experiments where they injected the fluid into healthy recipients, and they could not make anyone sick whatsoever. And they mentioned even in this paper that there was another site with a different investigators doing a similar series of experiments. They couldn't get anyone sick. And it turns out, like I said, there's over 90 studies just on the cold and flu contagion that essentially demonstrate that it is not a contagious illness whatsoever. And Daniel Roidas recently wrote a book to this effect called Can You Catch a Cold?, which I encourage everyone to read. He does also review some other non-cold related contagion studies. There are some, and I'll bring up one because it's kind of humorous from my knowledge. And this was an experiment done on people without their knowledge by the CIA. And there are many experiments like this, and they've been disclosed through Freedom of Information Act file dumps, and there are books by various authors that you can read and study some of these crazy experiments. So this one, they offered some, random men walking on the street a free ride with a prostitute. But what the men didn't know is that the prostitutes had chlamydia. Or actually, it was gonorrhea, sorry. The prostitutes all had gonorrhea, and they were symptomatic. And these guys, these Johns, basically had sex with them, on the government's dime. And then they followed the guys to see if any of them got gonorrhea, and none of them did. So that's kind of an example. I know that study is a little bit sexy and less scientific, but there are plenty of rigorous controlled studies done on this. None of them demonstrate that we can transfer illness from one person to the other. Despite whatever may happen at chickenpox parties.
How do you explain the Cutter incident?
What Cutter incident are you referring to?
The 120,000 kids that were infected with a not attenuated virus but a live virus. It caused kids to get tested positive for the virus. 40,000 kids. There were estimated 120,000 doses of the Salk vaccine that was produced by Cutter Laboratories. There were 40,000 kids who developed an abortive form of polymyelitis. And there were 56 kids who developed paralytic polio as a result of those injections. So if there's no virus, then how do you explain that?
Was this a vaccine?
This is a vaccine. Yes, but it contained a live virus.
Well, it allegedly contained a live virus, but is that the only thing it contained?
Oh, I'm sure it contained other things, but how is that?
So
How is that
the
live virus?
How do you know? Wait, let me finish. How do you know it wasn't one of those other things that caused the toxicity? How do you know there was a virus in there? How was it proven? I'll bet it was through a PCR assay. Steve, you got to go back to the beginning. You got to look, how was that thing produced? What are the ingredients in it? What's their toxicity? A lot of the things they use to grow viruses are very toxic to humans, right? We know this from all of the toxicity from vaccines. Right?
Yes, but there were 40,000 of 120,000. 1/3 of the kids who were injected developed poliomyelitis.
Yes, I'm sure they did, but poliomyelitis was originally caused by lead arsenate and then later by DDT. There's excellent research on that from Forrest Moretti. You could look at the moth.
I'm familiar with his work.
I'm not saying that there's definitive evidence, but
these people weren't exposed to DDT.
Correlative.
Dr. Kaufman, they weren't exposed to DDT. How did they get poliomyelitis?
Like I said, what was in the injections? You don't know. Maybe there was DDT in the injection.
We do. It was the standard
in developing countries today, Steve.
If it was because it was the difference in these cases.
You're talking about epidemiology. All you can do is make an observation. Lots of people died. You can't ascertain the cause. You can hypothesize it was the vaccine. You could hypothesize it's any one of the ingredients of the vaccine. You could also look at other data and see if there were any incidents.
So you believe that it's...
that's what epidemiology is. You observe and you ask a lot of questions. Then,
so do you believe that the most likely...
If you want to determine the cause, you have to do actual scientific experiments after that. Like at the beginning, I said, you can generate hypotheses from epidemiologic data because I knew that you would try to convince me with that, but it's not rigorous. It's not powered to prove cause and effect relationships.
What's the most likely explanation, Dr. Kaufman?
Well, I don't know
You
the
inject
data. I don't know
120,000
the
kids
data.
and 40,000 kids develop
Do you want me to
poliomyelitis.
answer?
Yeah,
Do you want
yeah.
me to
What
answer?
is the more plausible explanation?
Steve, if you give me all the data that there is on this and I read it, I can tell you what's most likely. But all I've got is a little bit of information that you said. How would I possibly know? You can't just determine causation willy-nilly. It's very, very difficult. It's one of the most difficult things to determine through experimentation.
Would autopsies have helped determine something?
Well, it gives you more information from which to generate hypotheses. Yes, absolutely. Because, like, for example, here's something you can do on autopsy data is you could see, are any of the vaccine ingredients in the tissue that's damaged, right? Because we do have evidence of that with other chemicals, like from other sources, like persistent environmental pollutants like dioxin. For example, you see them in adipose tissue, right? And then sometimes in liver tissue of people with liver disease. So you can get more information from that, but it's still observational data. It's not an experiment that you could definitively determine a cause.
Right.
Well, how about Barnstable County, the incident with COVID-19 outbreak in Barnstable County? Do you think that that would be like how do you explain that? How do you explain all those people? There were hundreds of people who attended an event. There was like several thousand people attended. There were about 400 people who got diagnosed with SARS-CoV-2, and they were gene sequenced.
Mm-hmm. They got diagnosed with a test, or they were sick with symptoms.
Now, they were sick with symptoms, but 133 patients. There were 133 patients got identified with the B1617.2, which is the Delta variant of SARS-CoV-2. 133 patients. Did that just happen by accident?
So, Steve, once again, when you have only observation, you can't tell what the cause of the sickness is. But here's what's the most
likely? Just give me the most likely explanation, Dr. Kaufman.
I don't have enough information to know. Like my recollection is that those people weren't even sick; that they just had PCR tests. But let's look at the data.
They had whole genome sequencing.
Let's look at the data in Italy because Michael Bryant has done really in-depth analysis on the epidemiologic data from Italy, right? Which was the first place outside of China that they allegedly, had a COVID outbreak, and he found many signals in the data that completely refute any story of possible spread. And then, of course, we also have all of the contagion studies never demonstrate that contagion is a real thing. So there's got to be.
How do you explain Barnstable County?
When I went to my grandmother's, 90th birthday party, family reunion, we all were waking up vomiting that night. None of us had a virus. We all ate the chicken. So they could have all eaten the chicken, Steve.
Yeah, but you wouldn't have had genetic sequencing of 133 people at the party yielding identical results.
If a bunch of people get exposed to the same poison and manifest the same symptoms, they're likely to express similar genetic sequences. So that's
as the SARS-CoV-2 virus sequence.
Well, that's not a real sequence, as I explained before. That's just, there's no way, all right, so answer this.
Right.
How do you know that sequence is from a virus? Where has that been shown? Do you know how many?
Because all of these people who get infected, I'll answer your question, okay? Because all of these people throughout the world who are getting infected and are having similar symptoms.
Only one person in that study, only one patient. How do you know he had a virus?
Wait a minute. In what study?
The Fan Wu original genome paper that we discussed.
I'll tell you why. I'll tell you why we know that.
One patient.
Yeah, I'll tell you why we know that.
How do you know he had a virus?
Because Because we've been able to replicate the genetic sequence. It's a novel genetic sequence, and we've been able to find that genetic sequence in people, in millions of people throughout the world.
So Steve,
That's how I know.
never find the identical sequence twice. Okay, but
Whoa,
the original,
whoa, where do you,
that's why they say they
excuse
have
me?
12 million variants, because every sequence is different.
No, because there are only about 30, in any given point in time, Dr. Kaufman, there are only about 30 or so variants that circulate and that exist at any given point in time. This is not millions of variants. There are, right now, at this moment, there are probably 30 different variants of SARS-CoV-2 that are prevalent in the United States of America, as an example. It's not 300 million, it's 30. That's a very, very small number.
All right, I'm gonna ask a different question, if that's okay.
Go for it.
From
Sorry.
Oysen Page, the promo vid for this debate showed Dr. Kaufman talking about mislabeling deaths and hospital and nursing home protocols killing people early in the pandemic. Could Dr. Kaufman please expand on those? And I'm not sure what they mean, because obviously those patients were sick, but I think I'm insinuating the protocols. Like, how do you expand on all these deaths?
Well, I mean, this has been, covered extensively, but it varied from place to place. Like New York was the place where they went crazy for ventilators and, put people on ventilators that were breathing on their own perfectly fine. In, various places in Europe, you had a lot of euthanasia type drugs, like hospice treatments for people with an acute illness. That were used. It's been extensively reported on. There's also a lot of acutely ill hospital patients were sent to nursing homes where they couldn't receive any treatment because nursing homes are not equipped to deal with that. So, there were a lot of different things. And then, of course, we had, some of the toxic pharmaceuticals that were put into the, COVID protocols that were reimbursed very well. And then, of course, ultimately the poison injections. So, this iatrogenic problem was, explained quite a bit of the illness and the brief, especially it was probably the major cause of that brief, short-lived mortality spike in the spring of 2020.
The protocols in the hospitals, basically.
Yeah, combined with the nocebo effect to a degree as well.
Another question here from one concerned world citizen. When a family all gets a cold around at the same time, what do you think caused it?
Well, this is, once again, an example of a straw man, right? Because I'm saying there's no evidence that there's a virus causing it. So I don't know definitively what causes it. I, of course, have my own hypotheses, but they're not tested scientifically, and I don't know, if they're worth describing. But I did say this a lot during, my interviews during the pandemic, and I think it's kind of an elegant way to think about it, because I purport it to be like the cycle of the leaves shedding from the trees, which we all enjoy every fall. And it's a renewal cycle, right? Because the leaves are actually the respiratory organ of the tree, because they have stoma underneath the leaf that the air comes in, and right, they breathe the carbon dioxide that we exhale, and they put out O2. But they also get air pollution coming through there, right, that they have to filter out because they don't want it inside the plant. And that makes the leaves, kind of gummed up by the end of the season, so they fall off. The tree hibernates, right? This is deciduous trees, obviously. And then in the spring, it grows new buds and leaves with, brand new clean stoma, and so that it can undergo its growth cycle for the year. So our breathing apparatus is our upper airway. So we're shedding the lining of the upper airway when we get the seasonal signal. We know that cold and flu season always starts in, the early winter, late fall, when the humidity and temperature drop below a certain threshold. That's been well established by observation. And so, that it's a natural renewal cycle. And then some people you have more gunk build up in their airway than others, right? If they live around pollution, if they have pollution in their house, like they live with smokers, for example, or other things, or they just generally live a toxic lifestyle, and other people that tend to be really healthy and take good care of themselves, they don't get colds nearly as often. Just because they don't need to, they're constantly undergoing renewal cycles.
But you didn't explain why a whole family gets it at the same time. This happened to my family just recently.
I did, because I said it was based on an environmental cue. It's an annual cycle. So all the family experienced the same environmental conditions, and so they had the same biological cycle commence.
You know, it's interesting that I was on vacation in Greece with my extended family, and a large number of people ended up with SARS-CoV-2 afterwards. All of a sudden.
How do you know they had SARS-CoV-2?
They did lateral flow assay tests.
Now has that been validated against the clinical population?
It has been validated.
No, it hasn't. It hasn't. It's never been validated, Steve.
Right,
To get an EUA approval, you don't need to validate it, but it's the step one for getting FDA approval.
So how is it that when I got sick with COVID symptoms and I took a lateral flow assay test, it turned positive, and when I overcame my symptoms, the lateral flow assay test turned negative, and my symptoms were consistent with SARS-CoV-2.
So what if you turned over a card from the deck and it was an ace, and then the next card was a 2? Would that mean anything?
No.
So if you have a random number generator and then you're interpreting it based on thinking it's a valid test, and then that's validating your belief that it's a valid test, that's just begging the question, Steve. It has to be validated first. Like, you have to have a clinical sample of people who they can find the virus in those people, and then they do the lateral flow test on those people, and also on another bunch of people that don't have the virus, but they look for the virus and then they're unable to find it, and then they do the lateral flow test, and then they compare the results of the virus, let's say isolation, because that's their gold standard, and compare that to the lateral flow results, and then they can determine the error. That experiment has never been done for any COVID diagnostic.
Do you know how a lateral flow assay test works mechanistically?
It doesn't matter how it works. This process of validating a diagnostic test is the same no matter what kind of test, what technology it works on.
We're getting close to when Dr. Kaufman has to leave. I'm going to ask one last question I think is a good one here from Jeannie, and it's, isn't it possible that we are confounding and therefore rejecting the bad science that was used to try and prove the existence of viruses with the concept of viruses themselves? Isn't it more likely that the means that were established to prove viruses were incorrect and fraudulent, but the existence of viruses still remain and demand a better scientific process to prove their existence?
Well, that's what I would call wishful thinking or promissory biology. So you're saying, yeah, of course viruses are there. I just know they are, even though none of the evidence supports it. But if we just had more technology or did better experiments, we'd prove it. I mean, it's true that you can't prove that something doesn't exist, right? We can't prove God doesn't exist. You can't prove that unicorns don't exist. You can't prove viruses don't exist. But you can look at the proof that's proffered for their existence, and there simply is none credible to be had.
Wow. I asked some friends of mine, and I said, is there anything that has been more scientifically validated independently all over the world than the SARS-CoV-2 genetic sequence for this single-stranded RNA virus? Millions of sequences have replicated this. And I asked them, and the answer they gave was, well, I think probably gravity is the only thing in science that has been more validated than the sequence of SARS-CoV-2. So
it's never been validated. Steve, you're using the wrong word. It's not validated. It's been repeated. and be something repeated. When you repeat something that's incorrect and it goes the same, that all that tells you is that you were incorrect to begin with. It doesn't tell you that you're right because you can repeat the same mistake and get similar results.
No, but these are not random results. I mean, 30, it was
I know they're not random because there's a whole
29,000
kit of
bases.
primers and
Okay,
a
you take
template.
four to the 29,000.
Steve, how do you know that any of the genetic sequences are from a virus? How do you know they're from a virus? They weren't taken from a virus.
Because they are taken from people who
Yes,
are.
they're taken from humans. So why aren't they human then?
Why aren't they human? Because they're a virus. Because they're a novel
How-
virus
how
that
do you
hasn't
know
been
they're a
sequenced
virus,
before.
Steve? How do you know they're a virus?
Well, they're not a bacteria because we would be able to see
How do
the
you know?
bacteria. Because
what?
we'd be. I just
Wait,
told
wait,
you
wait.
that. I just told you that because if it was a bacteria are larger than viruses. You
saying we
surely
can't
know.
see
You certainly
viruses?
knew that.
They're invisible?
No, and you can see them under electron microscopes. In fact, Tom showed you some pictures of that, and then you didn't get back to him. So how do you explain Tom's pictures?
Do you have
He
the
showed
paper
you.
here? Can you show me the pictures? I don't remember. This was a few years ago.
You did this 3 years ago. He showed you the pictures. He gave you the paper.
Can you bring me the paper? I mean, I know that
Refused to engage with him. Yes, of course.
Steve, if the
Here.
paper proved that there was a virus, do you think I'd still be here saying that viruses don't exist?
Yes, I think you would be. Yeah, because you didn't engage with him on the paper. He showed you the electron microscope.
Steve, stop
Images.
going there. The guy is like a teenager with name calling and insults. Okay, I'm not going to waste my time.
Wait. Now what you're doing is you're using ad hominem attacks rather than disputing him on the science. Why don't you just, instead of attacking him as a person?
You're absolutely correct. I apologize, but I don't engage in scientific discussions with people who use that kind of tactic. So that's why I did not have the discussion directly with him. But like I said, I did analyze the paper. I pointed out the flaws in it, and I published it. And you can go look at that. But if you don't know the actual procedure used for an experiment, I cannot really talk about it effectively. So the only question that is related to the genome is there's no way to know where any of those sequences came from because they never had a virus, took out genetic material from it, and sequenced it. They sequenced a human fluid that has a mixture of unknown organisms, and then they put the sequences together in a computer simulation to make a bigger sequence. And that sequence itself was made of 30,000 actual sequenced strands. And it's not known where any of those 30,000 come from.
That's not how it works. That's
Yes,
not how
it is.
genetic
It's right in the
that is
Fenwick
not how
paper that I read
that
earlier.
is not how gene
Steve,
sequencing
to know
works.
how it works, you have to read the papers. It's in the paper.
No, I don't know how
sent
gene
you. Go read
sequencing works, and it does
read
not
and
work the way
can-
you described.
You can see.
The culture had been sequenced.
You haven't provided the link for one paper. There's no way the audience can
Here,
check what
I
you're
can
talking
send. I
about
can.
and read the methods
Hey,
section. I put
sure.
all the
Here,
all
let
the
me,
papers on
Livio,
screen.
I'll email Livio the link to the paper right now.
All right, email it to me. I'll read it. I'll come back and talk to you about it.
can-
After I read it, if you like.
Great.
We can have a second version where we might be able to bring in some other experts, right? Since both of you have said not an expert in certain things, we can bring in some experts and have another conversation with more data, more experts.
Well, if we can bring in 1 million people, but to have a productive discussion, whoever participates, one has to either know the literature or be willing to listen and try and learn it, right? And there have to be ground rules about logic fallacies. We can't be using logic fallacy because it'll just derail any real learning or progress.
Yeah, we'll set up a proper format with rules and do it properly versus just an interview conversation. So I think that would be very productive to have something like that, if you're open to coming back, Dr. Kaufman, with.
Hey, I love having these discussions. And, I didn't expect anybody's mind to change today, but I think we definitely got a lot of information out there, in all respects. And, it's everybody out there can make up your own mind about what's true, what's not true, and about how you're going to deal with your health. And I think we all agree, stay away from any immunizations.
We definitely agree on that.
I think that's a great way to end on a really good, agreeable, stay away from immunizations and keep your immune system as healthy and strong as possible.
Your whole body.
Your whole body, everything, mind, body, and soul. Dr. Kaufman, I want to thank you for coming on tonight, and you are a true trooper, and I really appreciate you being here. And Steve, thank you again also for the conversation and just the tenacity again that you had to get what you want out of this conversation. and I appreciate you both, and hopefully we can have a different kind of round two, because I do think that all of us are on the right side as far as trying to help people get to the truth of things, and especially when it comes to the vaccines and the damage that they have done and continue to do. And that's where I lies to keeping us on the same side of that.
Absolutely.
Yep.
All right. And I know you have to go, Dr. Cobb. Steve, thank you. I'm going to finish off solo on the end. So thank you both, and have a great rest of your evening.
All right. Good night.
Good night.
Good night.
Everyone else, hang in there, because I wanted you to see the Nurse Angela speech from this past weekend. A little segment from it was great. I've been reading the comments, and I'll just leave it at that. I know there's a lot of opinions out there about tonight, and I appreciate you all for being here and sticking with us. And Steve's tampon, that's not cool. I'm just going to call you out right now, and I won't let you back in here for using that kind of language. Not cool. So we are a 501c3, and we are donating all the proceeds tonight to Cody, who is fighting for his life. So if you can and will contribute anything, will help Cody tonight. Text to VSRF at 53555. And I'm sure Cody and his family would appreciate anything you can do tonight. Also, coming up, Fauci rally on June 25th at 5:30 p.m. at Dominican University in San Rafael, California. We are hosting a Fauci protest rally. Please join Steve, Nurse Angela, and Lisa Leahy at a nearby restaurant at 4:30 p.m. Pacific Standard Time, again on June 25th. Doesn't say what day of the week that is. I don't have my calendar open, but June 25th at 4:30 p.m. for a happy hour hosted by VSRF. Tickets to the happy hour cost $150 and are tax deductible. We will put the link in the chat. Afterwards, we will head over to Dominican University and begin the protest. Fauci is due to speak with Barbara Boxer at 7 p.m. That event is ticketed and costs $48 to attend. Steve will speak at the rally, and he will be attending the ticketed event as well. We will put all the links in the chat. And also, if you want to go to our swag shop, you can donate by purchasing some of our great misinformation super spreader, VSRF, Defeat the Mandates swag that is in our shop. And then next week, we are working on who our guest is going to be, but we are going to have a special H5N1 deep dive into this avian flu. So join us next week, same time, same place here on our Rumble channel. And we're going to sign off tonight with a video clip of Nurse Angela's speech, again, as I promised, at the Reawaken America tour last weekend. And then I'll play a little bonus clip after that from JP Sears talking about or showing Marjorie Taylor Greene's grilling of Fauci in front of Congress last week. And thank you again all for joining us tonight. And we will see you all next week. Have a great evening.
How many of you here have been vaccine injured or know someone that has been vaccine injured? Wow, I am so sorry. Before I talk about the vaccine injured, I just want to share a little bit of journey of the tragedies that happened during the pandemic. This was truly a plandemic of the misinformed. Our world was shut down due to a virus that had a 0.35% chance of killing us. To put that into perspective, a bad flu season is about 0.5%. They told us to stay home till we were blue in the face and not get medical care when there was known safe and effective drugs out there that they hid from us. Once patients were placed in hospital systems, they were given deadly protocols that killed them. Our loved ones died alone. It was completely tragic. I am appalled by what they did to us. I'm sorry for all the lives that were unnecessarily lost. This occurred while we were locked down. Businesses collapsed. Fauci was at baseball games. I am from California. In California, we were locked down. We stayed home. I pulled my daughter out of school, not on purpose. I was forced to. While we were locked down in California, our governor, Gavin Newsom, was out having dinner with his friends and family, wining and dining at the French Laundry. Nancy Pelosi had her hairdresser open up her hair salon to get her hair done, while my hairdresser and thousands of others lost their businesses and livelihoods while we were staying at home. And our health director, Dr. Sundari Mays, who previously worked for the WHO and the CDC, she was telling us to stay home. Meanwhile, she was in bars, and she got her second DUI while we were locked up, and she hid that from us. I honestly can't make this stuff up. What happened next, in my opinion, is one of the most tragic things of the pandemic. It was crimes against humanity. They promised us a safe and effective vaccine that would end the pandemic and stop the transmission. I can tell you, they never tested whether it would stop the transmission. It was not safe. It was not effective. In fact, in the clinical trials, a child was harmed. Other participants were seriously injured and disabled, and none of us knew that. It was horrific. Americans were forced to take this vaccine to put food on the table. Children were bribed to take it with pizza parties, with donuts, with pizza money. I can tell you as a nurse, with informed consent, there should never, ever be bribery. College students were forced to take this to get an education. I am blown away that we allowed this tragedy to happen. Fauci lied to us. He told us so many things that were not based on science. The 6 feet apart, the masks, the one mask, the two masks, the masks that we had to wear in airplanes except when it was snack time. The restaurants that we finally were allowed to go to, but when we sat down, we could take our mask off as if there was some magic bubble that kept us safe. As a medical professional, none of this was based on science whatsoever. It was such a tragedy. We lost so many lives.
I think all freedom lovers want to see Fauci pay. And recently, at Fauci's Capitol testimony, Marjorie Taylor Greene plunged the sword of truth right into Fauci's filthy intestines. He squirmed. Take a look at this compilation video put together by Ultra Mega Party on Twitter.
Do you represent science, Mr. Fauci?
I am a scientist who uses the scientific method to gain information.
Yes or no? Yes or no?
No, that's not a yes or no
Yes,
answer.
it's a yes or no.
I don't think it is.
Okay, well, we'll take that as a you don't know what you represent.
Oh,
But this, as director of the NIH, you did sign off on these so-called scientific experiments. And as a dog lover, I want to tell you this is disgusting and evil, what you signed off on, and these experiments that happened to beagles paid for by the American taxpayer. And I want you to know Americans don't pay their taxes for animals to be tortured like this. So the type of science that you are representing, Mr. Fauci, is abhorrent. You confess that you made up the COVID rules, including
I didn't hear what you
6
said.
feet social distancing and masking of children.
I never said I made anything up.
You admitted that you made it up. You
I
made it
didn't
up
say
as you went.
I made it up.
So are you saying this is fake news, Mr. Fauci?
I didn't say I made anything up.
What did you say?
I said that it is not based in science and it just appeared.
But this is science. And making up guidelines like 6 feet distancing and masking of children. Do you think that's appropriate? Do the American people deserve to be abused like that, Mr. Fauci? Because you're not doctor. You're Mr. Fauci in my few minutes. No, I don't need your answer. That man does not deserve to have a license. As a matter of fact, it should be revoked and he belongs in prison.
Suspend.
Here you are without your mask, where you got to go and enjoy the game and sit right next to people not following the 6 feet of distancing, not wearing your mask, and everyone else was forced to stay home and stop enjoying life. And your science here, your science is displayed perfectly in this picture where children in school were put in plastic bubbles because of your science, your repulsive, evil science. And let's go back to your very own email. You said earlier you don't use email. Oh, you do. Right here. This is your own email where you said the typical mask you buy in the drugstore is not really effective in keeping out virus. I do not recommend that you wear a mask. This is your email. This is your own words. But yet children all over America were forced to wear masks. Healthy children forced to wear masks, muzzled in their schools. And then they were forced to learn from home because of your so-called science and your medical suggestions, while you and all your cronies get paid from Big Pharma. You know that what this committee should be doing? We should be recommending you to be prosecuted. We should be writing a criminal referral because you should be prosecuted for crimes against humanity. You belong in prison, Dr. Fauci.
That felt good to watch, and that weasel should be in jail.